students. Sri Manoj from MIT College, Pune. This is my fourth lecture on chromosomal theory of inheritance. In my previous lecture, we were discussing on the structure of chromosome, as well as the different types of chromosome based on the position of the centromere. This lecture is a continuity of the third lecture. Now, as you can see here, the classification of chromosomes based on the position of the centromere. There are four types of chromosome based on the position of the centromere. Amongst the four types, the first one is telocentric chromosome, second one is acrocentric chromosome, third one is submetacentric chromosome, and the fourth one is metacentric chromosome. Now, it is not necessary that you need to maintain the sequence of the types. You can start with metacentric and even end with telocentric. It doesn't mind. But where is the position of centromere in the chromosome is very important with respect to that type of chromosome. That you need not change. Fine. That means you can change the position of the types. It's not necessary that telocentric to be first. Okay. You can keep anything first and you can keep anything last. But the position of the centromere is very important in a particular chromosome. Okay. Now, let's start with the first one, metacentric chromosome. According to this diagram, if you can see, metacentric chromosome, in this the centromere is located in the center of the chromosome. That is, it is located in the median. So when it is located in the center, obviously this arm and this arm is divided into two equal lengths. That is, this arm and this arm. It's an equal length. That's what I've written. The centromere is localized approximately midway between each end, and thereby two arms are roughly equal in length. And in this metacentric chromosome, they take a V-shaped structure during anaphase stage of mitosis. Okay, so metacentric chromosomes, they represent a V-shaped structure because the centromere is located in the center. Am I clear with this about metacentric chromosome? Next is submetacentric chromosome. What is the centromere here now? Centromere is located on one side of the central point of a chromosome. If you can see here, this is the centromere. This centromere is located towards one side of the central point, towards the end of the chromosome. Centromere is submedian. This is submetacentric. Metacentric means median. So submetacentric will be submedian. So that in the centromere, which is located towards one end, it will give rise to two different size of the arms. One arm will be shorter, the other arm will be longer. Okay, so one is P arm, which is shorter, and other one is Q arm, which is longer. And which shape does it represent? It represents J or L shape. If you can see here, this is the L-shaped structure, or you can see here is a small letter J or J-shaped structure during the anaphase stage of the mitosis. Okay, so this is submetacentric chromosome. Now, what is next? Acrocentric chromosome. What is there in this acrocentric chromosome? Where is the centromere located? Centromere is located close to one end of chromosome. If you can see here, this is the centromere. It's located close to one end of this chromosome. The centromere, since it is located to one end of chromosome, okay, it gives rise to unequal arm length. Arm length is unequal. Arm means this is one, this is the other of one chromosome. Acro, in this acrocentric, refers to a Greek word means peak. It's at the peak. The centromere is at the peak. So it is called as acrocentric. Acro means peak. The P short arm is so short that it's hard to observe, but still it's visible. It is not visible, it is present. Okay, and which shape this acrocentric chromosome represents? Acrocentric chromosome represents a rod shape like structure during anaphase. This is about acrocentric chromosome, which is showing a rod shaped structure. Okay, next is telocentric chromosome. Centromere, which is situated at one end of chromosome, that is at a terminal position, is called as the telocentric chromosome. It lies at one end. And even this represents a rod-shaped structure during anaphase. So this is a rod-shaped structure in case of telocentric chromosome. Fine. So these are the four types of the chromosome based on the position of the centromere: metacentric, submetacentric, acrocentric, and telocentric. Okay. Now talk about X and Y chromosome, the human X and Y chromosome. Before we start with this X and Y chromosome, I would like to introduce some terms which will be highlighted when I'll explain you the structure of X and Y chromosome. Now, there are some regions present on the chromosome. Those regions are euchromatin and heterochromatin. Now, what is this euchromatin and heterochromatin? Now, chromosomes may be identified by regions that stain in a particular manner when treated with various chemicals. Chemicals means the dyes, the staining agent over here. So these chromosomes, they have got different regions on them, which is stained differently by using different stains. Several different chemical techniques are used to identify certain chromosomal regions by staining them, and they form chromosomal bands. So on the chromosome, there are certain regions, but the regions cannot be identified on their own. Some staining agent are required, some chemicals are required for recognizing those chromosomal regions. So those regions which are showing staining, means which shows different staining areas, those are nothing but chromosomal bands on the chromosomes. There are some regions where darker bands are formed near the centromeres or on the ends of the chromosome, that is near the telomeres. Telomeres are the end point of the chromosome, and we have studied this in the previous lecture under the chromosome structure lecture. Now, in this case, the darker bands, they are observed near the centromeres or on the ends near the telomeres of the chromosome, while other regions do not stain as strongly. The position means in some region they are stained darkly and in some region they are stained lightly. So they have got their own names. The position of the dark staining region are heterochromatic region or heterochromatin, and the light staining region are euchromatic region or euchromatin on the chromosomes. That means the darker and lighter regions, they are stained by using certain dyes or stains or chemicals which will give rise to certain chromosomal bands on the chromosome. Okay, the one which is darkly stained are heterochromatin, the one which is lightly stained are euchromatin. Fine. This is a small flow chart I can show. This is nothing much you need to learn over here right now. Now the chromatin, you know what is chromatin? Chromatin is present in the interphase stage of the mitosis in the cell division cycle. Now this chromatin is made up of DNA plus histone. Now this chromatin is of two types. One is euchromatin, other one is heterochromatin. Euchromatin is the one which is less condensed region of chromosome. Less condensed means they are loosely condensed region. Chromosomes are loosely placed. But when you say heterochromatin, the chromosomes are very compact, very tight regions, tightly placed regions of chromosome is heterochromatic. At the same time, this euchromatin, they're transcriptionally active, while heterochromatin, they're transcriptionally inactive. Now, why is so? Because the DNA are easily, the DNA are easily transcribed. The transcription takes place over here very easily because these regions are less condensed. They are not that compactly packed. Okay, but when talking in heterochromatin, the DNA, they're not that exposed. So the transcription is not that easily done over here. So this transcriptional, they are inactive. Now, heterochromatin is in turn divided into two types. One is constitutive heterochromatin and the second one is facultative heterochromatin. Now, constitutive heterochromatin are the regions that are always heterochromatic and permanently inactive with regard to the transcription. Means constitutive heterochromatin is the one which is permanently heterochromatic region and which does not involve in any transcription. But when talk about facultative heterochromatin, they lie between euchromatin and heterochromatin. Sometimes the euchromatin they are heterochromatin. That is regions that can interconvert between euchromatin and heterochromatin. That is facultative heterochromatin region called the chromosomes. Fine. Now let's have some more explanation on that, heterochromatin versus euchromatin. Chromatin can be divided into two regions, euchromatin and heterochromatin, based on its state of condensation. Condensation means chromosome are they tightly what to say? They condensed, they're tightly packed, or they're loosely held. That's what is the state of condensation. Most of the cellular chromatin is euchromatin, which has a relatively dispersed appearance in the nucleus. Means the chromatin in euchromatin, they are dispersed appearance in the nucleus. They are dispersed. They are not present in one particular place. They are scattered, you can call it as. It condenses significantly only during mitosis. Genes within euchromatin can be transcriptionally active or repressed at a given point in time. That's what you have studied before. That euchromatin is a region where transcription takes place very actively because the DNA is exposed the DNA can be transcribed very easily. Fine. Now, heterochromatin is condensed in interphase. But when talking about heterochromatin, is the area which is more condensed. Frequently, it's localized at the periphery of the nucleus. Usually does not contain genes that are being expressed and has late replication just prior to cell division. Just a little time before cell division, the replication takes place. And where is this heterochromatin region present? At the periphery of the nucleus. And it does not contain any gene that has to be expressed. Okay, that is about heterochromatin. Next, heterochromatin can be divided into two types. One is constitutive and other one is facultative heterochromatin. Constitutive heterochromatin is always inactive because it always remains as a heterochromatin. Facultative heterochromatin refers to the DNA sequences that are specifically inactivated as a result of development or regulatory event. Then sometimes this particular facultative heterochromatin, they can be interconvertible between constitutive and facultative. Fine. Now an example of facultative heterochromatin is a mammalian X chromosome. Facultative heterochromatin example is mammalian X chromosome. Okay. Now this is the structure of X chromosome and Y chromosome. What can you understand with your explanation? Just from the structure, nothing is labeled much over here. Now what can you see over here? Now here in X chromosome, you can see the X chromosome is longer than the Y chromosome. Yes, X chromosome is longer than Y chromosome. Let's see the next part. X and Y chromosome carry genes that determine whether a person is a male or a female. Female has two X chromosome, you know, XX chromosome. Male will have one X and one Y chromosome, and Y chromosome is much smaller than X chromosome. This is just a general thing, you need to know this. See the structure of X chromosome and Y chromosome. And structure Y chromosome is mentioned over here. Okay. Now the structure of X and Y chromosome. X chromosome of most organisms are straight, rod-like, and they are longer than Y chromosome. That should be a first point of difference between the two. Second, X chromosome have a larger euchromatin and a small amount of heterochromatin. That's peculiarity. If you talk about X chromosome, they have large euchromatin, so there are more amount of DNA, hence much genetic information they carry because they have large amount of euchromatin because they are genetically active, and small amount of heterochromatin genetically inactive. Euchromatin have large amount of DNA and hence much genetic information they carry. But when talk about Y chromosome, they contain small euchromatin. They contain small euchromatin and large heterochromatin. Hence, it has little genetic information, sometimes referred as genetically inert or inactive. What are they referred to as? Genetically inert or inactive. This is what about the X and Y chromosome with respect to euchromatin. So, euchromatin is more in X chromosome and euchromatin is less in Y chromosome. Heterochromatin is less in X chromosome, heterochromatin is more in Y chromosome. So wherever euchromatin is more, it carries large amount of DNA and hence it has got more large genetic information. At the same time, Y chromosome has got large heterochromatin, so less genetic information and they're genetically inert or inactive. Okay. Now, this you all know in as you have seen in the previous lecture. Autosomes and sex chromosomes. Autosomes, there are how many pairs in humans? I mean, how many pairs of chromosomes are there in humans? There are total 23 pairs of chromosomes in human beings. After 23 pairs, I'm talking about pairs. When I'm removing pairs, it will be 46. 46 chromosomes. So 46 chromosomes can be distributed as 23 pairs. Out of 23 pairs, 22 pairs are autosomes. And the last one pair is sex chromosome. X and Y is a one pair of sex chromosome. Okay, so autosomes, they are of the same size and structure. And that's the reason why they are homologous. But when talking about sex chromosome, one pair of this chromosome is X and Y. Okay, the X and Y chromosome, they are not homologous. X chromosome is much larger than Y chromosome and contains many genes. And Y chromosome has only small number of genes. That's the reason why X and Y chromosomes are not homologous chromosomes. But autosomes are homologous because they are of the same size and structure. In humans and other mammals, females are X's and males are XY. Okay, so this is what is about autosomes and sex chromosomes. Autosomes are homologous and sex chromosomes are non-homologous because they carry X and Y chromosomes. Fine. Homologous chromosomes. Humans have 23 pairs of chromosomes. 22 pairs are autosomes and one pair of sex chromosome. Already explained, that is number 23 is sex chromosome till 22 is autosome. Autosomes are chromosomes that do not code for gender. Okay, autosomes are the chromosomes that do not code whether it's a male or a female. It codes, the sex chromosomes are the one which codes for the gender. In females, they are all homologous because they are all X chromosomes. These are female chromosome, both 23. In males, the autosomes are homologous, but the sex chromosomes are non-homologous because they are one and one is X, other one is Y. X is longer, Y is shorter. That's the reason why sex chromosomes, they are in males, they are not homologous. Fine. What is this? Now this structure, I need to explain about homologous over here. So I have taken this slide. Homologous chromosome means of the same structure. They are seen in the somatic cell, nuclear, body cells. They are diploid. They contain a homologous pair of each chromosome. Means the somatic cell, they contain homologous pair of each chromosome. One pair is inherited from father, another from mother. Father means paternal, mother means maternal. Okay, this you all must be knowing. Homologous chromosomes are the same size and structure. They carry the same genes at the same loci. That's what is homologous chromosome. Means they are the same size and structure and they carry the same genes at the same loci. But the alleles carried at each locus may vary. Alleles at each locus may vary, but they carry the same genes at the same loci. Okay, now if you can see here, these are the banding pattern. This is a centromere position. This is a banding pattern. Okay, different regions are obtained over here because of the different chemical stains used over in case of chromosome. Now you can see 22 of the human chromosome pairs are homologous. The other pair, the sex chromosomes, are non-homologous. Now it is a gene loci present on the chromosomes. These are two homologous pairs. This is one pair, this is the other pair. Fine. What is this? This slide is again I need to show you about this 23 pairs of chromosome. Sex determination will be done in the next lectures, but just concentrate over here. 23 pairs of chromosome are diploid somatic cells. They are diploid somatic cells. N is equal to 22 of them are autosomes, which are homologous pair. One pair is 23rd is a sex chromosome. XX is a female and XY is a male. This all you all know. Now the X chromosome is much larger than Y chromosome. X chromosome carries many genes in the non-homologous region which are not present on Y. What they're saying here, X carries many genes in the non-homologous region which are not present on Y. Okay. Now, the presence and expression of SRY. SRY means what? What is the SRY gene? There is an sex determining region present in the, what do you call, the homologous part. That carries a particular gene. It's called as SRY gene on Y. On Y chromosome, it leads to the male development. Okay, so I want to show over here about homologous, non-homologous. So I took out this picture. Next, let's see what is the difference between X chromosome and Y chromosome. Okay, now X chromosome is a sex chromosome that occurs paired in female and single in male. Obviously, you know, it's XX chromosome in female and it is XY in male. Y chromosome is a chromosome which is normally present only in male cell. It is XY, so Y is only present in the male. Next is it contains a gene for female sex determination. This XX chromosome is for female and contains a gene for male sex determination. It means this YXY chromosome is for male sex determination. And X chromosome bigger than this male Y chromosome, but then this Y chromosome is smaller than the X. This bigger X chromosome it carries 155 million base pairs, while the smaller chromosome Y it contains about 59 million base pairs. Okay, now this X chromosome it represents 5% of the entire human genome, but this Y chromosome represents 2% of the entire human genome because size is different. X is greater, larger, longer, and the Y chromosome is smaller. Next, it contains more genes, about 1,000 genes. X chromosome contains more genes, and here it contains few genes, about 70 genes, Y chromosome. Next, what's a genotype? I'm talking about genotype. Genotype is different, phenotype is different. Genotype means genetic constitution. Phenotype means what? The physical external appearance of a particular individual is a phenotype, which I have studied in the Mendelian lecture. Now, this X chromosome will carry XX genotype and Y chromosome will carry XY genotype. Fine. Now, next is it does not contain the XY genotype, but that's the reason why this contains SYR gene. I told you it's a sex determining region. So, contains the SYR gene which is involved in development of the testes. Okay, which is present on the Y chromosome. SYR gene. Okay, that's all for today's lecture. Thank you so much.